Hello, my name is Elizabeth Blackburn, and I'm going to tell you about the discovery of telomeric DNA and of telomerase. When I first started working on this question, the ends of chromosomes, and these are chromosomes here, were known to be very important for protecting the genetic material, but nobody knew what was there. Now, it was known that every chromosome had a long DNA molecule that goes from one end of the chromosome to the other, and that long DNA, of course, carries all the genes. And every time that chromosome is replicated, the DNA has to be fully copied. But it was also known around the 1970s that the DNA replication machinery of the cell wasn't able to completely copy all the way out to the very ends of the chromosome. And so the prediction of what might happen every time a DNA replicated in order for a cell to divide would look something like what's depicted in this slide here, which would be that the chromosomal DNA end region would get shorter and shorter and shorter until eventually the cells would have such short chromosomes that they would be perhaps missing something from their ends and then not able to divide anymore. And that was even predicted on these theoretical grounds to be something that was called senescence, which had been seen in cells growing in culture, that they, in certain cases, could only go through a certain number of divisions and then they couldn't divide any further. But nobody knew why. And so, how to approach this problem? Well, I began it by taking advantage of a particular kind of organism. This is called Tetrahymena thermophila, and it's a single-celled ciliated protozoan. And you can see in this scanning electron micrograph here all of the cilia all over the single cell. And the particular reason for this choice of organism was that these cells have within them some very short linear chromosomes in high numbers. And so what was known before was that while nobody knew what was at the ends of the chromosomes in eukaryotes, cells with nuclei such as Tetrahymena, certain very short linear viruses that grow in bacteria, called bacteriophages, had the ends of their DNA analyzed. And in this organism, Tetrahymena, the very short chromosomes were about the same kind of size, and so I thought, well, I would try and look and see what was at the ends of these chromosomes, these mini chromosomes. Now, at this point, DNA sequencing methods had not been developed. And so, although this is hard for you to believe probably these days, but that was the case. And so what I had to do was to rely on a different kind of approach, and that was to really try to stitch together pieces of information about the ends of the DNA that I could obtain by using labeling techniques, radio labeling techniques, which would incorporate radio labels into the DNA, and analyzing by combinations of enzymes and chemicals that would cleave the DNA into little pieces that were dependent upon the particular building block bases of the DNA, and patch together the sequences, sort of stitch it together like a jigsaw puzzle. So, for example, I would cut the DNA up and see that there were, for example, little motifs in it, like this one here, where there was this motif of 4 cytosine residues in a row, CCCC, that's C4. And in fact, furthermore, I could tell from some of these analyses that there was a purine that is an A or a G, and then CCCC, and then an A or a G, because of the way I'd chemically cleaved this DNA up. And furthermore, by quantifying what was there, I could find that there were actually many repeats of this little motif at the ends of these short mini chromosomes. And so, putting it all together, I was able to conclude that these little tiny chromosomes ended in something that had really never been expected, and that was a sequence that I've drawn the strand which is the complement of the CCCC strand. It's actually TT, GG, and just repeated over and over and over again, perhaps 20 to 50 different repeats. Now the question was, how did these repeats get there? Now at this point, there were a lot of possibilities that you could draw on paper. And often in science, what you do is you take an observation and you put it into the existing body of information. You say, well, how can I explain my new finding given the context of all the information that's known? So we knew about DNA, and we knew about its replication machinery, we knew how it recombined. And there were certain well-worked-out rules by then for this. But these kinds of DNA sequences were not obeying those rules. And I'll tell you how they weren't obeying those rules in a moment. But the first thing I think that's interesting is that there comes a point when you're working in science where you know a lot of things, you're trying to fit your results into well-established principles, and then it comes to a point where it just won't fit in that box anymore. You just can't push it into that box anymore. And you have to say, well, let's entertain some other possibilities. Now, the kinds of observations, and I won't go through them, were that there were variable numbers of repeats on these small linear chromosomes. Sometimes there were 30 or so, sometimes there were 50, 70, there was different numbers of repeats. Well, that was already odd, didn't look like the bacterial viruses that people were familiar with. And then there were some other observations, one of which came from the extraordinary biology of these particular organisms, of which Tetrahymena thermophila is an example. The way it's got very small chromosomes is because it has a stage in its development right after fertilization when it chops its chromosomes up into small pieces, and then these telomeric TTG4 repeats would appear at the ends. How did they get there? This was not obeying rules that were known for DNA, where DNA was supposed to be copied from DNA or at least recombined with DNA that was very similar. You weren't supposed to have two new sequences suddenly joined together. And where did these repeats come from anyway? So I decided to look in extracts of the Tetrahymena cells and see if one could detect if there was some enzymatic activity that might be able to add this sequence, TTGGGG, over and over again, to the ends of linear DNAs. I didn't know what kind of assay really to use at this point. But I did know there was a stage in the life of these organisms when the very short chromosomes got created. And in fact, they were created by chopping up the DNA for stories which are... They were created by chopping up the DNA for reasons that are a whole different story. And then the telomeric, as we call them, repeat sequences, were being somehow put onto the ends of these DNAs. Often places where there were no such sequences there before. So how did such a different sequence get joined onto another piece of DNA? That was what we were trying to think about, answering the question, how did these repeats get put on? And so after a lot of trying different assay conditions, I finally found a mixture that was able to give me the first hint that something was happening, because I could see increasing amounts of this sequence being synthesized, apparently from nothing, in the test tube. And then at that point, I was joined by a new student in my lab, Carol Greider. And so the challenge now was to simplify down this reaction that we had going on in order to be able to see if there really was an enzymatic activity that truly was doing what these experiments had been hinting at. And so Carol was able to refine and strip down the assay to its bare essentials to get to this following point, depicted here. So we would make a DNA oligonucleotide, which is this colored bar here, made up of the building blocks you can see that looked like the end of a chromosomal DNA. And then by adding the extract of Tetrahymena cells right at a stage when they were known to be making new telomeres, because the chromosomes at that point in their life cycle were being chopped up and telomeres were being added. So I reasoned that they might be enriched for any such enzymatic activity if such a thing existed. So we made extracts from cells right at this stage and found just by adding simple salts and two simple building blocks of DNA, GTP and TTP, that in fact repeats were added to the ends of these DNA chromosome end mimics. So this was very exciting for us because this said, aha, this really would be a potential way of solving this problem of DNA's getting shorter and shorter, because now here's a way of making DNA get longer. So we had an enzyme activity which was working in the test tube and adding nucleotides that corresponded to the telomeric DNA sequences to the ends of chromosomes. And we had to name this enzyme, because we couldn't say tetrahymena-thermophila telomere terminal transferase too many times. And so there was a bit of a kind of discussion through the lab, and actually Claire Wyman in my lab came up with the name Telomerase, which we thought kind of made sense, because here was a telomere, the telomere, and then ase sounds like an enzyme, and we were thinking of polymerase, where, you know, a polymerase is something that makes a polymer. So we said, well, here's telomerase that makes a telomere. So we were happy to add this new word, which eventually made its way into the dictionary. Now, just to give you an example of the kind of way these reactions looked, what this is depicting here is an autoradiogram, which is a fractionated mixture of the reaction products, which have been labeled with trace amounts of radioactivity, and then they've been fractionated in electrophoresis in what's called DNA sequencing gel, and they start with just the very short DNA oligonucleotide, we call it a primer, and then the DNAs get longer and longer and longer, and you can see that there's this lovely repeating pattern, and that pattern is the 6 base repeats of the TT-GGGG motif being repeated over and over. And you can see that with time, more and more of this gets added to the ends of chromosomes. So this was a very visual kind of demonstration of this activity, and we could quantify it and do many experiments to try and understand its nature. One of the things we found out not too long after first finding this activity was that the enzyme actually had within it an essential ribonucleic acid component. And what this does is depicted here. So it had a ribonucleic acid component in it, which made the enzyme very sensitive to the enzyme ribonuclease, not what you'd expect if it were just a protein enzyme. And within this RNA, there was a sequence shown in blue here, which was the exact complement of the TT-GGGG sequence that we found was being synthesized in the test tube. So with such a very strong hint, we decided this really was the enzyme. And now the challenge was to find out if this really was behaving in cells as it was behaving in the test tube. Because what we were finding in the test tube was that a DNA, such as a mimic of a chromosomal end shown in black here, could be elongated by addition of nucleotides and copying the template. This was our model, and that would now make the chromosomal end longer. Was that really happening in cells? So the answer to this gave us a bonus answer as well. We were answering this question by making small changes in that blue sequence here in the RNA, and then putting RNAs with the changed sequence into a cell and asking if the changed sequence in the blue sequence caused a change in the DNA that was added. For example, if this templating mechanism, as it's called, were really true, then if we changed a particular C into a G, then instead of being copied by templated synthesis into a G, it would now be copied into a C. Or for example, if we changed one of those A's into a G, it would now be copied into a C instead of into its normal T. And in fact, that was what we were able to show in experiments that were done in cells by Guoliang Yu and John Bradley and Laura Artadi. Now that also gave us very unexpectedly another answer to this question. How do tetrahymena cells respond when telomerase is not working? We weren't actually initially setting out to do this experiment, but serendipitously, one of those changes in the template sequence, or in fact a couple of different ones, gave the sorts of effects that are shown here. So certain changes, for reasons to this day we really don't understand, would cause the enzyme, instead of simply synthesizing the corresponding repeated sequence with the corresponding mutation in the DNA that was copied from the template. Instead, these changes caused the enzymes to just choke up and it wouldn't work. It just basically stopped the enzyme working, as though it just sort of got choked up in its active site and can't work properly. Now that was very lucky for us because it allowed us to say what happens when an enzyme that doesn't work is present in the cell. Now what happened was that this telomeres started getting shorter and shorter, and then over the course of about 20 to 25 cell divisions, they progressively got shorter and shorter, and then the cells ceased to divide altogether. And so that told us something very important, which we can sort of summarize here. Tetrahymena cells are normally immortal. That is to say, they keep multiplying pretty much forever. However, all we had to do was to inactivate telomerase by this very small surgical strike in the essential RNA component of the enzyme, which inactivated the enzyme. And now the cells became, if you will, mortal. They could have a certain number of cell divisions during which their telomeres progressively shortened. They got too short, and then the cells ceased to divide. So loss of functional telomerase was leading to progressive loss of telomeric DNA from the chromosomal end, just as had been predicted from the original predictions of DNA replication if there weren't some compensatory mechanism. And so many cells have sufficient telomerase, and so they can now maintain telomeres at various lengths, but maintain them sufficiently well so cells can keep multiplying. Well, we discovered telomerase really by trying to answer a very basic question, which was how do chromosomal DNAs solve their problem of incomplete replication? And it was very much driven by, first of all, the idea that if there's some interesting problem in biology, you want to go for it and try and understand how nature is working. I think in the back of my head, I was always aware that nature tends to be very conserved in many of its most fundamental molecular mechanisms, as had been amply learned from the central dogma of DNA information going to RNA, going to protein, although interestingly this was a case in which a perfectly normal cellular enzyme was breaking the rule and RNA was being copied into DNA, something that people before had been thinking was only the kind of things that certain viruses and retro elements did, but they didn't realize until we found telomerase that in fact copying RNA into DNA can be a normal part of a normal cell's life. So we did think that this was probably fundamental and likely to be conserved throughout at least eukaryotes. I think what was unknown was really what the implications of this might be, and this has taken many years to work out and is still very much in the process of trying to be worked out as it affects, for example, what happens to humans, because we seem to live our lives with a sort of transition, if you will, between situations in which telomeres are shortening and situations in which telomeres are lengthening, and how that balance and how dynamics is all played out over human life is something that's very interesting and may well have implications for long-term progression, for example, towards certain disease states. And so I think what I take home as a message from this is that one really wants to understand how biology works by working at it in the most sort of curiosity-driven, question-driven ways, and not necessarily trying to ask the question of some application, but just simply trying to understand how things work, because I think we won't predict necessarily what the ramifications of that would be. That's certainly been the case in our adventure in working with telomeres and telomerase.